Good afternoon, everyone. My name is Fred Dobbs, and I'm the chair of the Department of Ocean and Earth Sciences at Old Dominion University. It's my pleasure to welcome you to the 29th CRC Roundtable, a monthly virtual seminar series. Our intention is to host targeted, inclusive, and informed conversations that match scientific advances and management needs. The webinars are designed for contribution, not consumption. And as a reflection of that intent, our two panelists have been allotted about 25 minutes total, to provide introductory remarks, allowing a full 30 minutes of discussion. This is all about us, the collective us, giving us a place to ask clumsy questions and to practice the bravery and humility necessary for building the future we want. These webinars are recorded and past seminars are available on the CRC website. I encourage you to review these, especially if you have missed some of the past discussions. If you would, please type your name, organization, and location into the chat box so everyone can appreciate the breadth of our webinar community for today's discussion. We also ask that you stay muted with your camera off while speakers are presenting. We always start these seminars with answers to the following questions. Why these topics? Why now? And why these speakers? Today's topics, first, a primer on seagrass blue carbon, and second, the effects of floods on water quality, clearly deal part and parcel with Chesapeake Bay restoration concerns and should be considered in the formulation of management and policy decisions. With respect to why now, please know, participants, you are subjects in a roundtable experiment. CRC Executive Director Denise Wardrop and I decided to showcase research ongoing at the CRC's member institutions. In this regard, you should know that I represent Old Dominion University to the CRC's Board of Trustees and serve as the chair of that board. It made sense to Denise and me, therefore, that ODU should be the guinea pig in this endeavor. In future months, I anticipate you will hear about Bay-relevant research at the CRC's other member institutions in alphabetical order: Johns Hopkins University, Penn State University, the Smithsonian Institution, University System of Maryland, Virginia Institute of Marine Science, and Virginia Tech. And to the third why, let me tell you about today's panelists. Both are full professors in ODU's Department of Ocean and Earth Sciences. Both have strong research programs in the Chesapeake Bay and elsewhere in the United States, other countries, and in some cases out in the deep blue sea. Facilitating their research programs are a bevy of undergraduate and graduate students. Both Dick and Margie are important contributors to the educational mission of our department and of ODU more generally. Now for a little bit about each panelist in the order they will speak. Richard Zimmerman leads ODU's Bio-Optical Research Group. He earned his PhD in biological sciences at the University of Southern California. His research interests include the ecological physiology of marine photosynthesis, radiative transfer and remote sensing of optically shallow waters, ecosystem productivity, and numerical modeling. Margie Mulholland is the principal in ODU's Marine Nitrogen and Carbon Biogeochemistry Group. She was awarded her PhD in biological oceanography by the University of Maryland. In addition to nitrogen and carbon cycling, she studies harmful and other algal blooms, organic matter cycling, and nitrogen fixation in the marine environment. Speakers, I will gracefully let you know if we bump up against time. If you have material you are not able to share during the time limit, we'll find a way to make that information available to others afterwards. Attendees, please write your questions in the chat portion of this webinar. If we don't get to your questions during the webinar, we will give the speakers time to address them afterwards. We will then share that information back to the community. With that introduction, I'll turn things over to our first speaker, Dick Zimmerman.
All right, thank you, Fred. Let me get set up here. Hold on just a second. Oh, I need to share my screen first, sorry. All right, can you all see that?
Yep.
All right, great. Thank you. All right, so thank you, Fred, for that introduction. Just going to clear a couple of things off my screen there. All right, so I'm going to tell you a little bit about some of the work we've been doing in the Bay to assess the role of submerged aquatic vegetation in blue carbon. I'm talking for both myself and my colleague Victoria Hill, who is helping lead this project. And of course, the work I'm going to present today also involves collaboration of ODU students and colleagues at other institutions, and funding, of course, comes from the mostly from NASA through their ocean biology and biogeochemistry program. Okay, so blue carbon is a term that was coined about 15 years ago to emphasize the role of aquatic vascular plants in the carbon cycle. This includes marshes, mangroves, as well as seagrasses. And particularly for seagrasses, one of the problems is that global estimates of their importance is highly uncertain. And this is due to the fact that less than 10% of SAV resources have been mapped quantitatively across the globe. And this is partly due to the fact that mapping submerged plants is technically and logistically challenging. SAV abundances and distributions are also temporally dynamic on short timescales, which means that they need to be mapped frequently in order to get good quantitative estimates. They're also spatially dynamic on small scales, which means you need high resolution imagery in order to do this. And getting to carbon assessments requires some knowledge of the species composition, because the allometric equations used to get from biomass to carbon are highly species dependent. Now, we're fortunate in the Virginia area, here in the Chesapeake Bay area, to have access to the VIMS SAV aerial survey program, which has been working now for almost 40 years to provide the longest continuous record of SAV distributions and relative abundance anywhere in the world. And here's just on the left, you see a map showing all of the sites that they image. And on the right, you see a time series of SAV abundance, starting from the nadir period in 1980, when SAV abundance in the Bay was at its lowest, up to the present day, which is around upwards of almost 40,000 square kilometers. Notice, however, that this line at the top represents the SAV goal for restoration. So we're quite far away from restoring SAV abundance in the Bay to what is desired. Nonetheless, these data that have been produced by VIMS are critically important for management and science, and they've been used extensively for both activities. Nonetheless, these aerial survey methods face increasing challenges that are threatening the long-term viability of this data set. Number one is that the frequency and the scale of surveys is limited. They can basically do image the Bay once a year. And this is a result of operational costs for aircraft and scientists, as well as the logistic complexity involved in scheduling aircraft to collect the data. The efforts are labor intensive due to scheduling, ground support, and then manual photo interpretation of the results. And then the uncalibrated digital photographs that they get from this effort are not really amenable to automated machine learning techniques to help streamline some of these processes. And because they're uncalibrated, it means that they're affected by the illumination effects that change the relative brightness from one scene to another, which means that retrieval of absolute abundances is often challenging from these kinds of data. And since we can't get absolute abundances, getting to estimates of blue carbon quantification becomes challenging. So we've been looking at recent advances in satellite remote sensing to see if we can improve our ability to map SAV distributions across the Bay through time and get to absolute quantitative estimates. We've been working with a corporation called Planet Labs, which is now operating more than 200 CubeSats currently orbiting the planet, producing a global daily coverage of the entire Earth's landmass. Now, it doesn't cover the entire global surface. It doesn't do most of the open ocean, but all the land mass is being covered, including most of the coastal zones. The new satellites that they're launching include 8 spectral bands, so they're similar to spectral coverage with MODIS and VIIRS, the NASA satellite systems. They provide 3-meter ground resolution, which is right where we need it to be for mapping small patchy distributions of things like SAV. They provide a radiometrically calibrated image, which means that we have better reliability comparing from one sensor to the other and through time. They've harmonized their data sets to the European Space Agency Sentinel, which has improved atmospheric calibration, atmospheric correction. So they provide a product that's ready for us to use, just downloaded from the website. We have to do almost no additional modification to the imagery before we can use it. So our goal is to see if we can use these satellite remote sensing data to replace or at least augment the aerial surveys and develop an automated process to assist in the classification, and then generate quantitative blue carbon estimates for biogeochemical modeling purposes. I should note that these data are freely available for research purposes, and all you have to do is go to the Planet website and get an account, and you can start to do some of this stuff on your own. We're going to be looking at 4 different regimes in the Chesapeake Bay that we're working at. We're starting at the highly turbid oligohaline upper bay, which is dominated by freshwater vascular plants, down to the moderately turbid mesohaline central part of the bay around Smith and Tangier Island, which is sort of a transition between the freshwater and the saltwater domain, the polyhaline York River, which is dominated mostly by seagrasses, and then the oceanic coastal lagoons, where they have been an extensively restored meadow of Zostera marina, has the highest salinity, lowest turbidity, and some of the healthiest seagrass meadows in the region that we've been spending a lot of time working on. Okay, what we're doing is using a convolutional neural network system to classify the images into various categories. We can separate land, sand, marsh, optically deep water, and submerged aquatic vegetation in the image. And right now we've been spending a lot of time using the imagery to train the data, train the machine learning classification model, but we're on the verge of being able to have sufficient training in the model that we're starting to be able to give it images that it has not seen before and for which no training is provided, and it's starting to give us good classifications. And this is key to making this process automated, because this will remove the human step of having to provide training data. And then once we have the SAV, we have a physics-based model that will get us to leaf area index and blue carbon. So I just want to show you a couple of examples of what we're getting. Here is a satellite image, a Planet satellite image of the seagrass meadow in South Bay. The dark area here is the seagrass meadow that has been restored. And when we run our classification algorithm on it, you can see in green that the seagrass meadow pops out quite readily. In addition, we get very good characterization of the marsh, bare sand areas, optically deep water where we don't see a bottom signature, as well as land and the beaches. Furthermore, if we overlay the percent cover polygons that the VIMS seagrass mapping efforts have produced for the same time period for 2021, we see really good coherence between what our automated classification scheme is able to pick up and the polygons that are drawn by VIMS, particularly for the higher density percent covers, 40 to 70 and greater than 70. We don't do as well in the lower density coverage, and that's simply because they have higher resolution aerial photographic imagery that they can see smaller patches that don't get picked up in our 3-meter data sets. However, if we just look at the total area for the eastern shore over there, we see that the area that we have been mapping from satellite imagery compares very favorably with the total area that's being produced by the VIMS polygons. And you'll notice one other thing: each of those blue dots represents a satellite image that we have collected out of their archive between 2018 and 2022. In contrast, the estimates from the VIMS polygons are based on annual surveys taken once a year. So you can see another feature of the satellite imagery is we're starting to fill in and get time series measurements that become really important if we start to look at other patterns of variability in these systems through time. And then once we have the images classified, we can use the brightness of the SAV pixels to get an estimate of leaf area index here, which is total leaf area, that we can then use allometric equations to convert to total carbon. So we are getting to absolute estimates of abundance and carbon that we can then now start to convert those areas into total amounts of carbon. And here you see the time series that we've estimated for the South Bay in gigagrams of carbon through time over the last 3 years. You see there's been quite a bit of variability, but more or less stability. It hasn't changed very much. And the average density out there we estimate it to be about 40 megagrams of carbon per square kilometer in the seagrass meadows. We've done a similar thing for Goodwin Islands National Estuarine Research Reserve. Here you see the Goodwin Islands map. The top panel shows the VIMS polygons drawn for 2021. The middle panel shows our classification, our pixel-based classification for the areas. You'll see some differences here, particularly in the lower density classes where the VIMS mapping is showing much more extensive areas of seagrass meadow than we are picking up. In fact, we are discussing that with them, whether some of these things are actually true meadows or we're actually measuring seagrass or we're talking about habitat. So we're having a conversation about how things are done because different approaches produce slightly different results. The meadows here in Goodwin Islands are much smaller and less dense than in the South Bay. It's about a square kilometer around Goodwin Islands. And the standing plant biomass is lower, obviously, for that reason. And the carbon density of the seagrass meadow is also lower in this region.
Nick, you're at 12 minutes.
Thank you, Fred. Finally, I'll take you up to the Susquehanna Flats, where we're starting to see an area where we get dramatic seasonal differences in SAV abundance. Here again, 2 satellite images, one from January showing the extensive mudflats on the Susquehanna Flat and some areas of vegetation that we've picked up with our analysis. Then in August, at the height of the SAV abundance, you can see that the entire flat is completely vegetated. And not only is there submerged vegetation there, but it's so dense that there's actually emergent vegetation. And doing the same kind of analysis we've been doing on the South Bay, we're starting to get estimates of total area for these things that we hope to eventually be able to convert into estimates of blue carbon resources for these different environments. Now you notice there's a big gap here between January image and the summer images, and that's simply because during 2022 there were a lot of clouds and we weren't able to find any reliable data for that period. So even though there's a lot of satellites up there flying, clouds are still a problem and they will always be a problem for remote sensing. So let me just conclude by saying that we've got a new generation of satellites that's greatly improving our ability to assess blue carbon in these systems. With daily image acquisition, we eliminate the logistical problem associated with tasking satellites or aircraft, and it's starting to enable time series observations that are not otherwise possible doing it from the ground. Having radiometrically calibrated and atmospherically corrected data allows for the training to be the machine learning algorithms to be automated because we're getting them trained to a point where they can perform consistently across time and space. This also gives us quantitative values for standing SAV biomass and blue carbon rather than just percent cover. And the thing I'd really like to emphasize is that the satellite technology is improving rapidly, almost day by day, meaning that this is the kind of technology that's available not only to us, but this is something that can be scaled across the entire globe. And we're working with colleagues to push this out into the Gulf of Mexico, up along into other areas along the Atlantic coast, Pacific coast, and even out into remote Pacific islands. Thank you.
Thank you, Dick. Folks, please, we're going to hold off on questions until both speakers have presented. Now we're going to segue to our second speaker, Margie Mulholland. Great, we can see your screen, Margie, but we don't hear you.
That's because I have to unmute myself, turns out. Okay, there we go. Okay, now can everybody see that?
We can see it and we can hear you.
Okay, so today I'm going to talk about water quality and coastal flooding and sea level rise. And I have to thank, first of all, some of my co-authors. Alfonso Macias Tapia really ran this whole show, and Derek Loftus at VIMS helped us with the calculation of floodwater volumes. Okay, so most everyone in this audience know that the Chesapeake Bay is under restoration, and that impairments have been linked with human activities, including point and non-point nutrient sources. And these impairments make the news here in Hampton Roads region. And this is a figure just showing the front page of our newspaper one day when there was an algae bloom that was bioluminescent and created quite a stir. A little background on the Chesapeake Bay restoration. You probably don't need this, but the original restoration agreement was signed in 1985, but it was non-binding. And the restoration showed insufficient progress in 2010. And so the EPA stepped in as a result of consent decrees in Virginia and Washington, D.C. And essentially what came to pass was total maximum daily loads were set for the entire bay. And these included segment-specific TMDLs for particular segments of the bay. And the regulated sources included point sources as well as non-point sources from runoff and atmospheric deposition. So what are we missing from these loads that we're counting? This is the nutrient diet. The TMDL is the nutrient diet that the Chesapeake Bay was put on. How are we cheating on the diet? We're counting the runoff, but really at this scale, I don't think so. So those are some pictures of Hampton Roads when it floods. But in this region, we flood even when the skies are blue, when there's no rain associated, and that's just because of the high tides. This is a figure from Tal Ezer just showing the hours per year above mean high water. And this goes from 1930 to 2018. And you can see that this rate of flooding has increased over time. And when it floods, it certainly is inconvenient to get around. Our cars go underwater. We can see in a lot of these pictures here where cars are being flooded. On land, we've started adapting in the Hampton Roads region. There are signs telling us where not to park, telling us not to do wakes while driving down the road, no wake zones. And we have meter sticks sometimes on some of the roads to show just how deep the flooding is so we don't pass through it. And again, warnings about where to park. Most of the efforts to adapt or to mitigate for a sea level rise have focused on the landward side of the equation and protecting infrastructure. We're raising houses, we're not occupying flooded buildings and roads, trying to raise things and keep people off them. But our group, we started thinking about the water side. All these drains go into the Chesapeake Bay, and as the water comes up, it brings back pollutants into the water. And this is one of Alfonso's cartoons, essentially showing when there's tidal flooding, how the waters are in the estuary here. We get flooding, it inundates the landscapes, and then it puts everything back in the water as the water returns. And this can include construction debris and all sorts of waste. Really anything you can possibly think of gets in that water. So other things going in are things like hydrocarbons, oil. You can see the sheen on these waters, and they're all going back into the Chesapeake Bay when the waters recede. We have litter. You can see all sorts of junk. We have a contest in our lab to see who can take the picture of the grossest thing that's in the water. There's a couple places there are always trash cans tipped over here. This one's wrong for so many reasons. It's not only a recycling bin, but it's filled with trash, and it's sitting in the water. And we get everything. This is water bubbling out of a drain cover, and there's toilet paper coming out of it. And here is some pet waste. And here's the pet that was emitting that waste. So everything's going into the water. Why do we care? Because these nutrients going into the bay are not quantified and are not being counted towards the TMDL. And we're hoping to restore and protect the bay, both from these harmful algal blooms, as well as other ecosystem impairment. And how can we hope to restore the bay if we're not even counting the nutrient loads in the floodwaters? In fact, maybe that's why the bay got another deep D+ this year from the restoration. So how do you quantify the nutrients going in from coastal flooding? It's actually pretty tricky because you have to have hundreds of floodwater samples in a particular region to be statistically valid. And we need to focus a lot of these measurements in a single watershed, and that watershed needs to have a TMDL that we can compare it to. Then we need to collect these samples nearly synoptically at near high tide. So we need lots of samples and lots of people to do this. And so we need to pick a time that we know it's going to be flooding in advance so that we can plan. And then we have to be able to calculate the floodwater volumes based on the extent of flooding. We have good models down here in Hampton Roads that can do that. And we need to know what was already in the estuary prior to its encroachment onto the land. So how did we do it? We tapped into a citizen science project called Capture the King Tide. And this project is aimed at mapping and building awareness in the public of coastal flooding. And essentially a nonprofit group, Wetlands Watch, developed a phone application. And it looks like this. It's called Sea Level Rise. And people download it onto their phones. And when it floods, they walk the water line and drop pins that are GPS points that go back to a data assimilation model. So what we did was we tapped into this project. We thought of a catchy name, and we called it Measuring the Muck. And we engaged students, in particular, AP Environmental Science classes from local high schools that needed volunteer hours. And we trained them to collect water samples while also mapping. And we had to find an accessible watershed to work in. And we chose the Lafayette River system because it's both close to ODU. It's a small watershed with its own TMDL. It's centrally located with lots of Catch the King volunteers. It experiences lots of tidal flooding. This is a picture of it with, I think it's 1 foot of sea level rise superimposed on it from a model that's available in Norfolk, the city of Norfolk. And we have time series station in this river that we collect in-water measurements with great frequency. So what we did, the volunteers signed up for Catch the King. They were trained with those volunteers, and then we trained these students to also collect samples. We provided them with sampling kits, then deployed them to areas they knew would flood, brought samples back to the laboratory at ODU, and processed them there until we could analyze them. We sent out these students in groups because sometimes we had young kids, and so we put them with someone experienced, with someone inexperienced, and we were able to collect about 200 water samples in each year, and also help the Citizen Science Project that, by the way, won a Guinness Book of World Records, largest citizen science project. And we provided them a physical log to log in samples, and they also could do so on the phone app. Okay, and we could map our results onto different areas to look for hotspots. I don't want you to focus on this map. This is actually for ammonium. But what I want you to focus on is our floodwater calculation, because to actually calculate that nitrogen load, all we needed to know was the volume of floodwater, and we could get that from Derek's models. And we needed to know the median floodwater concentration, because that was conservative estimate. And we subtracted out what was already in the river. When we first did this for 2017, and this is just for nitrate, we calculated it was about 1,265 kilograms of nitrogen, translating to 2,784 pounds of nitrogen from nitrate alone. In different years, we've been doing this since 2017. We just finished our 6th year of it. And you can see that we collected lots of samples. We had varying numbers of volunteers, but the height of the floodwater really varied in the king tide, which is the astronomically highest tide per year that we conducted these events. But the inundation volume could really vary. And so the floodwater volume varied from year to year. But keep in mind this 2,784 pounds of nitrate from nitrogen, because when we look at the Lafayette River's TMDL, and we look at the nitrogen load allocation, the annual load allocation based on runoff, we see that it is just 1,941 pounds of nitrogen per year. So we just delivered the entire annual load during a single blue sky flooding event. And again, this is just the load based on runoff. So we know that flooding can be variable and that the land prehistory can be highly variable. So we suspect all loads are not equal. And so we also established sentinel sites. So we could assess the seasonal variability, the different landscape prehistory, and different land uses. So we had different sites, parking lots, roads, trashy areas, residential areas, etc. And from there, we were able to calculate in this left side panel that based on the mean high water level, the difference in the nitrogen from the estuary, floodwaters to the estuary, or the load, increased with the level of mean high water over our sentinel sites. And these were just all pooled. Same with that for P. We can see a high variability, but generally an upward trend. And when we backcast this using water level data from Sewell's Point, we can see that this net DIN load over time has really increased just due to the fact that tidal flooding has really increased. And this is true for both nitrogen and phosphorus. Now, on another separate note, we were also measuring, we had the Virginia Department of Health and the Hampton Roads Sanitation District were our major sponsors, and they examined fecal contamination in a subset of our samples. Each year we had about 40 samples that were analyzed. And in this figure, you can see this box and whisker plot showing the Enterococcus concentrations in the different samples. This red dotted line way down here, that is the Virginia Recreational Water Use Standard. That's 104 NPN, or most probable number. And you can see that very few samples met this. Most of them were way higher this, and many of them greater than 24,000, which was the high end of the scale. Above that, we could not detect. So there was a huge amount of fecal contamination in these waters. And when we have flooding down here in Hampton Roads, we've had days where kids go to school late or not at all because buses can't get to their stop. Kids treat these flood days like snow days, and they go out and play in it. So I would argue, based on this fecal contamination, we don't want to do that. And so to conclude, climate change and flooding are going to jeopardize our coastal restoration efforts, and we need to adapt our efforts to not only think about resources on land, but also reduce water quality impacts. And with that, I'll say thanks, and thank again the Army of Volunteers that helped with this, and the Hampton Roads Sanitation District and the Virginia Department of Health. The end.
Thank you very much, Margie. That was great. I have some questions for you personally, but I invite the attendees to send in questions on the chat. You see that Macon Thompson has put that instruction out for you. Okay, well, my first question is to Margie. In fact, Margie, as I understand things, you worked with the Lafayette River, a single river. What are the possibilities and obstacles to extrapolating to larger sections of the bay? Can you take this technique and make wild ass guesses about what's going on elsewhere?
What we're trying to do is actually just that, is we've really been impeded by the lack of funding. The Hampton Roads Sanitation District has been our primary funder. And what we're trying to do is get into other branches initially of the Elizabeth River, because that's tractable, it's near to campus. We have a large volunteer group there. And there's the wider variety of land uses will hopefully enable us to characterize a load by land use or by at a tributary level. And the idea would be, if we could do that, then we could scale up with models to bay-wide forecasts for that. And so we're working on that.
Thank you. And Denise Wardrop, Executive Director of the CRC, has a follow-up question, I think, in this regard. The amount of loading is alarming, but what management efforts could be taken for source reduction? And I would add, don't they already exist?
That's a great question, but not really for runoff and whatnot. They exist, but really there's not much in place for when there's a tidal flooding event. You know, what needs to happen is maybe we need to target street sweeping for before that happens. Have projects like down here, there's a River Star program that has citizens, businesses, and industries has them try to tend to their landscape so they reduce their loads to the Elizabeth River. but people don't even really get warned sometimes about the tidal flooding in these areas. So they're not checking the tide chart, so they don't see it coming. So at a minimum, send out alerts, because especially during spring tides, when we know it's going to flood, or at a king tide, we could certainly send out alerts so that people can, move cars, do things that would clean up the landscape a little and help keep things from going in.
Thank you. And Dick, we're going to move over to you. If I remember the graph correctly, you were showing, I believe it's carbon biomass. Maybe it was coverage of seagrass being relatively in agreement between the VIM survey and yours. And over the relatively short time period that was shown, what do you project for the future? You showed the goal way up top there. The future with respect to warming, increased sea level rise, and increased CO2 concentrations in the atmosphere. You're muted, Dick.
Thanks, Fred. So that plot showing the agreement between our estimates of aerial coverage and VIMS was for the coastal lagoons for South Bay and Spider Crab Bay out there, which are probably the healthiest SAV meadows around. Maybe those and the ones up at Susquehanna Flats in the freshwater area are the healthiest. So the relatively stable numbers that we're getting for there are suggesting that the restoration effort is at least in South Bay, it's getting close to a stable point. It's not going to be. Although it's expanding into other regions in the coastal lagoons, in that area that we're studying now, it's starting to become pretty well maxed out, I think, as inhabiting most of the areas where their seagrasses are allowed. It's moving north and moving south, but we didn't include that in our area. So that's the stability factor in that region. The values for the entire Chesapeake Bay are still very, very low, and the water quality in the bay is not nearly as good as it is in the outer coastal lagoons, which is why it's less abundant around Goodwin Islands area that I showed you in that. And as for the long-term prospects of SAV, that's at this point, it's highly debatable. There are people arguing that the temperature rise is going to basically cause the plants to be extirpated. But some of the work we've done on CO2 stimulation and photosynthesis suggests that at least some of those temperature effects negative temperature effects may be offset by CO2 subsidies. And so our arguments are that the plants are probably able to survive warmer temperatures than we think if we can improve water quality. And I think that's the key is survival of the SAV is critically dependent upon water quality. So all of that message that came from that Margie was pointing out about need to control nutrient loading, that goes right to the question, your question about what's going to happen to SAV in the future. It's largely I think it's going to be more linked to what we do about controlling nutrient loading and those problems we've been dealing with for 30 years, and that climate change is going to be a secondary factor.
Mm. And let me follow up with a question from Catherine Kreider to you, Dick. Do those summer algal blooms caused by nutrients decrease the quality of the satellite imagery with respect to the seagrass visibility?
Actually, I've got an image I can show you if you want. Let me just share is that okay? Can I share my screen and show you an image?
Sure. Make it quick.
Yeah, quick. Let me share my screen and pull it back up. Just happen to have one in there in case somebody was interested. Where is it? Here it is. Okay, can you all see that?
Yes.
So this is a PlanetScope image of the York River that we collected on 26th of August. And all of this ribbons of dark you can see coming down the river. This is a Margoliphodinium bloom coming down the river. Down here in the bottom corner, let me get my...
Dick, if I could interrupt. So Margoliphodinium, for folks who don't know, is a single-celled dinoflagellate. You're looking at a plankton bloom, phytoplankton bloom.
So down here is the seagrass area around Goodwin Islands that I showed you in my talk. And you can see that the bloom is heading right for it. And of course, what the bloom does is they compete with light for the seagrasses and large, dense abundances of phytoplankton are one of the major contributors to the loss of SAV in the bay. So if this mass of harmful algal blooms were to come down and completely surround Goodwin Islands, yes, on that day, on that image, it would probably very much compete with our ability to see the SAV underneath the harmful algal bloom. But I just would like to point out that these daily images that we're getting from the satellite mean that we can now start to track the blooms. We've got 2 weeks of images that we've downloaded around this period of time, and you can see the slow progression of the bloom down the York River and out into the bay. So
Nice.
it not only can we use it for the SAV, but we can use it for lots of other things too.
Yeah, great. That's great. I hadn't seen that image. Great. Margie, over to you. Bill Ball asks, does Norfolk have separate stormwater / sewer systems or combined? And for either case, what is being done to eliminate sewer overflows during storm events? And a related question, are check valves being placed to disconnect stormwater from the tributary waters during king tides?
That's a good question, and I believe they are combined. We're a really old sewer system. And
Actually, we're mostly separate.
oh, we are. Okay, I
Yeah,
didn't know that. Okay, so yeah, so I know they've been working on it because a lot of our storm drains, when it floods, is the water comes, that's how the water from the estuary oftentimes gets onto the land. And so we'll have areas that seemingly aren't near the coast. There's one intersection near downtown that it's not anywhere near the water, but it's a low spot. And so it always floods because water is coming up and out the storm drains. And again, right now, mostly the efforts are to protect infrastructure on land. And so the placing of check valves and whatnot, I really don't know what they're trying to put in place, but the problem is so systematic around the city that I don't know how you really do that for all these storm drains. That water comes from the estuary into the streets from those. I don't know how you would check all those particular valves.
Yeah, I know in my neighborhood, there is what is essentially an experimental project that I've heard is being looked at throughout the state, where check valves are being put in to keep the high waters from coming onto the land. Let's see, several questions here. Back to one that actually was sent in earlier. Margie, can you talk about trends in heavy metals, for example, mercury, and the effects of storm runoff on that?
That's another thing we would love to expand into. And in addition to expanding our scope, we would love to expand our suite of analytes, but we are unable to analyze those, especially given we've been doing this on just, like $10,000-$20,000 a year to try to do what we can. But so we would love to expand that. It's just a matter of money and facilities and someone to make those analyses.
Yeah, and then in an entirely different context, a question to both of you, and Macon, I think you have some input on this after Margie goes. Are you aware of any BIPOC, that's Black, Indigenous, people of color, led environmental groups in the Hampton Roads, Virginia community? Margie, if you'll speak to that first, and then Macon, chime in if you would.
Sure, yeah, in our high schools, we actually have chapters of the Surfrider Club, their groups, and the Norfolk Public Schools is predominantly minority. And so there's a lot of leadership there with those high school clubs, and those are clubs that we've been recruiting for Measure the Muck. There are also some efforts at Norfolk State that we're trying to partner with them in the eastern branch of the Elizabeth River because they're doing some monitoring on the eastern branch that will give us important in-river monitoring. And then if we can, again, get funding to expand to some of these other tributaries, that's our plan, is to engage groups and communities where they live. There's a lot of civic leagues. There have been a few flooding projects done in underserved neighborhoods. So we're we're working on that. I think Macon, you had something to add to that as well.
Yeah, thank you, Margie. About a year ago, I interned with the Great Dismal Swamp Stakeholder Collaborative, and they're based out of the Hampton Roads, Norfolk area. They're really tied to the Great Dismal Swamp National Heritage Area and do a lot of environmental outreach, education, and also a lot of historical acknowledgements. They're led by a 3-person leadership team that represents historic Black communities as well as Indigenous groups. The Nansemond Indian Nation is very involved with them, and they also reflect the interests of the local environmental community there. And they're striving for a National Heritage Area designation to further expand environmental and historic protections on that site. And I'll drop the link in the chat to their website if anybody wants to connect with them further.
Thanks very much. And Macon Thompson, by the way, attendees, is the technical wizardry that's putting on this show. Thank her and Greenfin Studio more generally for the tech work on this. I appreciate it. Dick, Denise Wardrop asks, what are potential plans for using this carbon information? Can this be part of restoration plans that satisfy multiple goals, climate resiliency, habitat?
Yeah, yes, absolutely. One of the important things that globally the blue carbon community wants to do is use this to make estimates of the contribution that these ecosystems make in terms of the carbon cycle. Partly, I think, as a justification for continuing to restore and protect these habitats. It's another ecosystem service that these systems are providing, and quantifying those services is an important part of that goal. So that would be a practical application for it. But there's also a lot of fundamental science questions that we have about these systems. How much carbon do they actually store? One of the things I didn't talk about today due to lack of time was the fact that there's also a below ground reservoir of carbon that is even more poorly quantified than the above ground reservoir of carbon. And there are lots of interesting questions there about what's going on with that system, because one of the things that we've noticed, we know about these systems is that although they're producing lots of biomass, if you actually look at the amount of carbon in the aquatic sediments, that's very, very low. They're around 1% carbon by weight. So one of the big questions is, if we're producing all this biomass and supposedly burying a lot of it in the soil, where's the peat? And we actually have a proposal pending at NASA right now to try to investigate some of those things. We have some ideas about what's going on with that organic carbon and where it might actually be going.
And when you talk about underground or below ground carbon, you mean roots and rhizomes? Is that
well,
right?
yes, plus buried leaves and other allochthonous carbon material that gets trapped and buried in among the seagrass leaf material, basically litter.
Yeah. Okay, thank you. Margie, from Rohit, how effective is the civic engagement from efforts like yours, together with the Sea Level Rise app, for educating and engaging the public? I'm wondering how to engage the most people in the fight against climate change. You mentioned that you were underfunded, but how have you been able to reach out to the public? You're muted, Margie. You're muted.
Sorry, I muted myself because someone is leaf blowing behind my house. But that's a great question, and I'm really excited about that, because this Catch the King effort was made possible, and that was going out and doing the flooding awareness during the king tide in the fall. But that was made possible because of our local media outlets in the Hampton Roads region. A former environmental reporter at the Virginian Pilot, Dave Mayfield, he got the idea and went to WHRO, our public media outlet, which has an extensive education arm, as well as Wavy 13, a news channel, and the Daily Press and the Virginian Pilot, our 2 local newspapers. And they all made a push in the first year, advertised. They got a great logo with Elvis, and they reached out to the community. And like I said, it won a Guinness World Book of Records record for the biggest citizen science project, because they were able to recruit hundreds of volunteers through the relentless media blitz that went on about it. And there were people from here, really statewide, that were out there mapping and getting trained to map. And so what we did is we went into the schools, the high schools, the public schools, and we recruited from the environmental clubs, the specialty programs, and the AP Environmental Science. And then we went back and also made presentations to all these groups afterwards. So we were able to get in the classrooms. The kids were excited because they'd heard about it on the news or from their parents or people talking, and they were just happy to get out there. In fact, in one of the years, because we just didn't have the space to accommodate, we had to turn people away even, because it was such a success. And then the pandemic happened, and that whittled our numbers to where we just didn't advertise because we couldn't handle more. But really, that effort from the former environmental reporter, and engaging the public media outlet in particular, because they've persisted in sponsoring it publicity-wise for all 6 years what we've been undertaking it. And so having their engagement and their large educational focus there has been fabulous for getting out the word and making sure people know. And now just scaling up becomes a problem of space, money, and space.
Space, money,
and money and space.
Dick, I see that you answered a question from Steve, but perhaps you can restate that and answer it in voice.
Yes, this was part of what I think you had asked a few minutes ago, but Steve wanted to know about what do we need in terms of water quality measures to extract the seagrass abundances. And primarily what we need is estimates of chlorophyll and suspended matter to reliably extract the SAV abundances. And some of that we get from local sondes, in situ sondes that are adjacent to our sites. And some of it we're now starting to be able to estimate from the imaging itself, much like NASA does to derive chlorophyll concentrations and backscattering and suspended matter from their satellite imagery. Basically, it's a very similar process.
Yeah, good. Thank you. And Margie, back to those high school students. Has there been any follow-up with the participating high school students on shifts in their attitude or intention to pursue environmental career opportunities or post-secondary studies training in that area as a result of participating in your project? The all-important pre-post work that you have no money or people for, but go ahead.
But still, that's so funny that you should ask, because Alfonso Macias Tapia, who's my graduate student, he got a Knaus Fellowship and is now up in the NOAA Education Office. And we were talking about how to pull together a manuscript with to talk about the citizen engagement and how has it changed these student lives. And so we're actually working on a survey to look at volunteers. Many of the high school students graduate and move on, but some of them participated in multiple years. And so what we're trying to do, we're both scientists, but he has some people he can access in the NOAA Education Office. And I don't know if you're an expert in doing this, but we would like to design a follow-up survey that we can deploy with these volunteers to see how it's influenced their career choices. Because the students let me tell you, they get so enthusiastic about it, and they love being partnered with the graduate students as well, to get that near peer sort of thing going. So if you have any ideas on developing survey instruments, Alfonso and I are sort of muddling along trying to learn to do that and work with some of his current NOAA colleagues to do that, who have some ideas on it.
Yeah, that's one of the great syntheses that can occur between the egghead researchers like us and real educators who know how to do this thing properly, such that it's well quantified. Good, thank you. Bill Ball asks you, Dick, whether other satellites under NASA's control might be able to be entrained in getting images for you.
Well, we can use their algorithms to help get at some estimates of water quality in the area, because the bands that these commercial satellites are covering are very similar to the NASA ones. But the big problem with the NASA satellites is that they're not, the spatial resolution is not good. Now, Landsat's getting close at about 30 meters, But the ocean color satellites that NASA operates, VIRS and MODIS and the new PACE program, they're still, the pixel size is on the order of, the minimum pixel size is on the order of 300 meters, and most of them are around a kilometer, which means that most of our areas would be basically 1 or 2 pixels in their satellite image. And they tend to stay, for the ocean stuff, they tend to stay farther away from shore where a lot of our things are, because they get adjacency effects from the shore as well. So what we really get from the NASA folks is that they spend a lot of time developing algorithms that are even for very useful in the Chesapeake Bay, because it's a unique optical environment. And I think we can just port those algorithms right over to these commercial satellites that are giving us the higher spatial resolution that we need to map these SAV meadows that are really close to shore, and in the small tribs and other complex habitats that that NASA's just not capable of working with.
Thank you. Great. I'm going to start the wrap here, and I will ask you, Margie and Dick, you can choose the order, about what you'd like to leave. This is essentially the elevator speech, such that people will remember to tell family and friends over dinner tonight what it is that they heard as you presented your seminar.
I think Denise asked a great question at the end, is should we be focusing on these shallow water environments, given the importance of them? And I would argue for a different reason, that these are the environment people see. And so that I think it is important to work in these areas and do our public outreach in particular in these areas, because this is what people see. They don't see the blue waters, the offshore waters even, or much beyond where their line of sight goes if they're looking out at the ocean. So I think these are incredibly instructive areas for people to engage, and then work with people on the bigger picture and the planetary bits and pieces that affect our climate.
Thank you. Dick?
Yes, I agree completely with what Margie just said. I've often remarked that if you really want to know how far people care about the ocean, just go to the shore and throw a rock. And about as far as you can throw a rock out is about as far as most people care, which encompasses most of the areas we're working with. I would like to leave everybody with one thought about the satellite stuff, though, is that the technology is improving rapidly. And what used to be the domain of experts in ocean optics and satellite data and remote sensing, these images are now becoming routinely useful in a common format that virtually anybody with a PC and a little bit of interest can start doing. So I encourage all of you to go to the Planet Labs website. You can get an account for free, and for personal research and academic purposes, they have a very generous data sharing programs. The software to interrogate their websites is pretty easy to use, and there's lots of freeware out there available to start working with this stuff. So here's an area where labor and costs are really becoming not an issue. As long as you've got a personal computer, you can start to do some of the satellite imagery analysis that I was showing you today.
Great. All right, everybody, head over to your computers. I guess you're already on it. All right. I want to thank Dick and Margie very, very much, and all the attendees, and thank you also to Greenfin Studio for the tech work. The next CRC Roundtable will be on the 15th of February. We look forward to seeing you then. Thank you all very much for attending. Good afternoon.